Begin bacterial genetics with horizontal gene transfer. Horizontal gene transfer means bacteria receive DNA from another bacterium, instead of passing genes from parent to offspring. This is different from normal inheritance, where a bacterium copies its own chromosome and divides. Horizontal gene transfer is clinically important because it allows bacteria to gain antibiotic resistance or virulence genes very quickly. For example, a bacterium that was previously sensitive to antibiotics can suddenly become resistant after receiving a resistance gene from a nearby bacterium in a hospital setting. When a question describes resistance appearing suddenly or spreading rapidly between unrelated bacteria, horizontal gene transfer is the correct framework, not slow mutation. On exams, the strategy is always the same. You identify how the DNA moved, what form the DNA was in, and what factor allowed or blocked the transfer. The question is written so that only one mechanism fits all clues. If one detail does not match, that answer choice is wrong. Transformation is the uptake of naked DNA, meaning free DNA floating in the environment. This DNA comes from bacteria that have lysed, which means their cell wall broke and they died, releasing their contents. Only competent bacteria can perform transformation. Competent means the bacterium has special surface proteins that allow it to bind and pull DNA into the cell. The classic organisms tested are Streptococcus pneumoniae, Haemophilus influenza type B, and Neisseria species. These organisms are grouped together because they are naturally competent. After DNA enters the cell, it must recombine, meaning it must integrate into the bacterial chromosome. If recombination does not occur, the DNA is destroyed. A critical exam clue is DNase. DNase is an enzyme that breaks down DNA. DNase only destroys naked DNA in the environment. It does not affect DNA inside bacteria or inside phages. If adding DNase stops gene transfer, the mechanism must be transformation. For example, a lab experiment may show that bacteria acquire resistance unless DNase is added. That finding proves transformation. Another common stem describes bacteria gaining a capsule after exposure to heat-killed bacteria. Heat-killed bacteria cannot divide or make pili, but they still release DNA, which again points to transformation. Conjugation is DNA transfer that requires direct physical contact between bacteria. This contact occurs through a sex pilus, which is a protein tube that connects two bacterial cells. Conjugation always involves plasmids, which are small circular pieces of DNA separate from the chromosome. An F plasmid is a fertility plasmid that contains genes needed to form the sex pilus. A bacterium with this plasmid is called F positive, or F positive. A bacterium without it is called F negative, or F minus. During standard F positive to F negative conjugation, a single strand of plasmid DNA is transferred, and the recipient becomes F plus. Importantly, no chromosomal DNA is transferred in this basic process. This detail is frequently tested to eliminate incorrect answers. An advanced form is the HFR cell, which stands for high-frequency recombination. In an HFR cell, the F plasmid is integrated into the bacterial chromosome. When conjugation begins, DNA transfer starts in the plasmid region and continues into nearby chromosomal genes. Because the mating bridge usually breaks early, the entire chromosome does not transfer. The recipient stays F-, but it may gain new chromosomal genes. Exam questions often describe chromosomal gene transfer but emphasize that the recipient remains F-. That combination specifically identifies HFR conjugation. Transduction is DNA transfer mediated by bacteriophages, which are viruses that infect bacteria. There are two types. Generalized transduction occurs during the lytic cycle, when the phage destroys the bacterium. During this process, bacterial DNA is broken into fragments. Sometimes, random bacterial DNA is accidentally packaged into a phage head. When that phage infects another bacterium, it injects bacterial DNA instead of viral DNA. Because this is random, any bacterial gene can be transferred. If a question mentions a lytic phage and random gene movement, the answer is generalized transduction. Specialized transduction occurs during the lysogenic cycle, where phage DNA integrates into a specific site in the bacterial chromosome. If excision is incorrect, the phage removes adjacent bacterial genes along with its own DNA. Only genes near the insertion site are transferred, so this process is not random. This mechanism is tested heavily through toxin production. Many important toxins are encoded by phages, including diphtheria toxin, cholera toxin, Shiga toxin, botulinum toxin, and erythrogenic toxin. If toxin production appears only when a phage is present, the mechanism is specialized transduction, also called lysogenic conversion. Transposition refers to the movement of transposons, which are DNA segments that can move between DNA locations. Transposons are often called jumping genes. They do not need matching DNA sequences to move. Transposons commonly carry antibiotic resistance genes. They can move these genes from the chromosome to plasmids, which then spread easily between bacteria. A classic example is TN1546 carrying the VANA gene, which allows vancomycin resistance to move from Enterococcus to Staphylococcus aureus. When resistance appears suddenly across species or involves multiple antibiotics, transposition is the most likely mechanism. To tie everything together, examiners rely on consistent clues. If DNase blocks transfer, naked DNA is involved, which means transformation. If direct contact is required, think plasmids and pili, which means conjugation. If phages are involved, decide whether the transfer is random or toxin-specific to identify generalized or specialized transduction. If resistance spreads rapidly across species, especially with multiple drugs, think transposons. Quickly recalling what we just learned, transformation is uptake of naked DNA from lysed bacteria. Only naturally competent organisms do this. Classically Streptococcus pneumoniae, Haemophilus influenza, and Neisseria. DNA must recombine with the chromosome. DNase blocks transformation, which is a decisive clue. Heat-killed bacteria can still donate traits. Tested uses include capsule acquisition and antibiotic resistance without cell contact. Conjugation requires direct cell-to-cell contact via a sex pilus and always involves plasmids. In F-positive to F-negative conjugation, only plasmid DNA transfers and the recipient becomes F-positive. No chromosomal DNA moves in standard conjugation. HFR or high-frequency cells have the F plasmid integrated into the chromosome. They transfer chromosomal genes, but the recipient remains F-positive due to interrupted transfer. This explains rapid hospital resistance spread. Transduction is gene transfer by bacteriophages. Generalized transduction occurs in the lytic cycle due to packaging errors and transfers random genes. Specialized transduction occurs in the lysogenic cycle and transfers specific adjacent genes. Many toxins are phage-encoded, including diphtheria, cholera, Shiga, botulinum, and erythrogenic toxin. Toxin presence dependent on a phage indicates lysogenic conversion. Transposition involves transposons, or jumping genes. They do not require homology and often carry multiple resistance genes. Transposons explain multidrug resistance across species, such as VANA on TN1546 moving from Enterococcus to Staphylococcus aureus. A quick summary of the decision rules: if DNase blocks gene transfer, the DNA must be naked in the environment, which identifies transformation. If gene transfer requires direct cell-to-cell contact and involves pili, the mechanism is conjugation. If a bacteriophage is involved and the transferred bacterial genes are random, the mechanism is generalized transduction. If a bacteriophage is involved and the gene transfer is toxin-specific or depends on phage presence, the mechanism is specialized transduction, also called lysogenic conversion. If multidrug resistance appears rapidly across different species, especially in a hospital setting, the mechanism being tested is transposition.